So in many ways, you can think of my talk as following on Adina Roski's talk yesterday, which I thought was super interesting in thinking about, like, how do we construct our ontology in neuroscience? How do we find the sorts of concepts and ideas in neuroscience among all the possible ones of how we could represent what's going on in the brain? And so this is joint work with Emanuele, Ralph Edos, and a bunch of others at the University of Iowa, and really credit to Emanuele, who's done much of the heavy lifting on this. What I'm interested in is how do we find the right granularity of representation for the causal relation between what's going on in the brain and what is happening in behavior. So we might have very fine-grained, high-dimensional measurements of what's going on in the brain. We might have very fine-grained, high-dimensional measurements of the resulting behavior. And now the sort of question is, at what sort of granularity should we locate the causal relation that connects what is going on in the brain to that of behavior? And so the question is, of course, both on how coarse or fine-grained should we capture the features of the brain, and so how coarse or fine-grained should we capture the features of the effect. And my notion of causation that I'm going to use here is actually exactly what Lauren said at the end when pressed on providing the definition. I care about causation in the sense that I can intervene on it, and I see a result of that intervention. So I'm not going to concern myself, at least in this talk, about circuits of mechanisms or some sort of detailed pathways that are there. For those of you who are philosophers, this sort of picture might look reminiscent of that about mental causation, where you might ask about what exactly is the relation between mental effects and the brain states underneath. I'm not going to concern myself either today with mental states. I'm going to really look at features of the brain, coarse features of the brain, and ask what sort of causal effect do we have on behavior. And specifically, in the end, what I want to look at in terms of a concrete application is I want to take lesion maps of the brain that we have from the collaboration with people at the University of Iowa, and how well people with these lesions performed on a whole variety of cognitive tasks. And I want to ask, given that sort of data, at what sort of level of granularity can we say something about the causal relationships between the types of lesions and the effect on what types of behavior those lesions might act? That's the overall goal. But first, I want to sort of start by developing quite abstractly the sort of question of finding the right granularity of a cause-effect relationship. That is, suppose we have some micro-level space at which we have fine-grained measurements of both the cause and effect, and we want to ask how coarse can we go in this regard. Now, one thing that just off the bat I think should be obvious, but I want to state it nevertheless because it does happen a lot when people look for coarse causal relations, is that you might think what we should do is dimensionality reduce the cause and dimensionality reduce the effect, and then once we've done that, learn the causal relations between those two dimensionality reduced spaces. I think that's a terrible idea, and it's quite obvious, I think, because, of course, what the sort of salient features in the cause might be completely irrelevant for the causal relation to the effect, and similarly features that you would get rid of if you just dimensionality reduced in one of these spaces might actually be important and subtle for the causal relation, even though they're not important for the actual, Uri, you're making me nervous.
Let's see. Sorry. We could not see your title.
Oh, I'm sorry. I'm sorry. Okay. Okay. So I don't think we want to do any sort of identification of the causal relation or the granularity of the causal relation by looking at dimensionality reducing each of these things independently. So what I think we have to ask ourselves is that there are two types of questions. One is, given effect of interest, what is the right granularity of the cause that we should consider as cause of that effect? And then similarly, the converse, the effect of cause question, given the cause of interest, at what level of granularity should we consider the effects of the cause? And now, given that you might be starting from fine grain on both the cause and the effect, I think you want to ask both and ask how coarse can I go? So just as a concrete sort of example to keep in the back of your mind, so suppose I have detailed information about the education history of a whole bunch of individuals, and I have detailed information about their income 10 years out after graduation, right? Then I might say that, okay, look, for this sort of causal relation, I don't need a lot of the detailed educational history. I just need to know what type of bachelor they did, namely, was it philosophy or neuroscience, right? Then that's a huge effect on what sort of income you have 10 years out from graduation. Yes, I can tell you about that. But then similarly, you might say, okay, well, that's the appropriate level of granularity for the causal relation here if I'm interested in talking about the cause of future income. But then you might also say, okay, given that I have on the cause side, I can give you information about what type of bachelor I have, then you might find that with regard to this relation, detailed information about the income 10 years out from graduation is perhaps not the relevant granularity on the effect, but in fact the effect should be coarsened as well, namely what really only matters if it's greater or less than 60K. That's all perhaps that our data really supports in terms of what sort of information we can say about the causal relation between the two. So this is the sort of question that I want to ask with regard to brain activity on the left-hand side on the cause and behavioral activity on the right-hand side for the effect. How coarse can we go, and how should we identify at what level we should stop? Now, given that I'm talking about causation, I obviously want to be sensitive to the fact that causation can be subject to, or causal variables can be subject to confounding. That means there could be a common cause that I may or may not observe of the two variables in question, and so I have to ask myself: is this sort of coarsening that I'm engaging in, is it just going to identify features that are actually confounded as opposed to key features that are causal? And so I'll come back to that and say something about how we might go about distinguishing the causal effect from that of confounding, even when we're searching for the right level of granularity. Okay, to motivate how we're going to address this question of finding the right granularity, I think it's very useful to have this sort of toy example that Spurtis and Shines developed in their paper in 2004. Many of you have seen this before, but I still think it's enormously helpful for illustrative purposes. Now, for all of those who know how cholesterol actually works, remember this is a toy example, this is not medical advice, right? It doesn't quite work like this. But suppose total cholesterol is made up of HDL and LDL, happy lipids and lousy lipids, right? And happy lipids have sort of a positive effect on heart disease, and lousy lipids have a negative effect on heart disease, expressed here with interventional probabilities. Then you wouldn't want to aggregate HDL and LDL up into total cholesterol. Why? Because the causal effect of total cholesterol on heart disease would be ambiguous. Namely, it would depend on the ratio of HDL versus LDL. You have over-coarsened this sort of system. You should have stopped in terms of your aggregation at the level of HDL and LDL, because that's where you had definite effects on heart disease. In contrast, if HDL and LDL had exactly quantitatively the same effect on heart disease, then of course it would be perfectly appropriate to coarsen up to total cholesterol, because both HDL and LDL, the distinction with regard to the causal relation on heart disease between HDL and LDL simply doesn't matter. We can coarsen the distinction away. Of course, with regard to a different effect, the distinction between HDL and LDL might still matter. I take this sort of criterion of identifying causes that have definite effects to be crucial in terms of identifying the appropriate level of granularity. And in taking that criterion as crucial, I'm in no way the first to identify that, but this has long been thought as the right kind of answer to at least addressing the cause question, namely our cause of effect question was, right, given an effect of interest, at what level of granularity should we consider causes of the effect? I think there's a straightforward answer to that. It was already made by the philosopher Stephen Yablo many years ago. And Jim Woodward, as Lauren also mentioned earlier, adopted this as well, is that among causes with definite effects, select the one that is coarsest, and this corresponds to what is known as proportionality in the philosophical literature. What I think is important to point out here is that, okay, you want to sort of start from a fine grain, and you want to aggregate and coarsen your cause as much as possible with regard to the effect, but and the sort of admissibility criterion on what sort of causes you're permitted to use is in terms of, does the cause have a definite effect? Does it have sort of, is it not a mixture of two different effects? And then because you want to avoid ambiguous manipulations, but then which one of all the ones that have definite effects do you select? Well, you select the coarsest one, because if you selected any finer one, you would still have definite effects, but you have redundancy in your representation. That's the sort of thought. So I think in many ways we therefore have a unique answer of what the appropriate granularity is for a cause given a particular effect. Is you coarsen while you have definite effects, and then you stop before you end up having ambiguous ones. Okay, we can formalize this all theoretically. We've done this in several papers. So the basic idea is that you would want to merge states in the cause space variable that have the same causal effect as defined by the intervention distribution. Of course, you have to sort of ask yourself, is that, okay, how do I identify the intervention distribution? All that sort of thing depends on the particular setup where we might apply it, and I will come back to how I do that in a specific example later. Okay. But that leaves us still with the effective cause question. How should we select the appropriate granularity of an effect given that we have a particular cause? So here, I think Lauren mentioned this as well, which was sort of a great setup for this talk, and as Jim Woodward suggested many years ago already, that really what you want in the effect is you want to be as specific as possible while retaining control. Okay, so if you have some sort of cause, you want to sort of keep the granularity of the effect as fine as possible without having sort of redundancy in it, or because you want to capture the full complexity of the causal relation that is there. So that means that you might start from, so one way to think of it is start from an extreme coarsening where of your effect, and then refine it make it as fine as possible while you're still retaining control over that sort of effect. That then selects the appropriate level of granularity to describe your causal effect between the spaces that you have here. So the answer, and I can give you all the detailed sort of theory for this in the discussion afterwards, or after per email afterwards, is that the general idea is that among the effects for which the cause has definite effects, select the one that is finest, that has the finest sort of granularity, because that way you retain the maximum representation of the causal relation between the cause and effect space that you're considering. Okay, again, we have this sort of nice feature then that the admissibility to what sort of effects we consider in a causal relation is one of definite effects again. So we want to have a sort of, it shouldn't be a mixture, but then among the ones that have where the effects are definite, we select the finest. And why is that? Because if you actually coarse grain from the effect that you selected while you have a definite causal relation, then you retain the definiteness. So you have this sort of duality between the how to select the appropriate grain of the cause and how to select the appropriate grain of the effect by, on the one hand, looking for proportionality for the coarsest representation of the cause that retains definite effects and the finest representation of the effect for which you have definite effects. This now means that you really have a unique level for any, if you have your data and your measurement, you identify in this way a unique level of the right granularity of representation. And moreover, this sort of level that you identify in this way remains subject to empirical revision, right? So if you have a lot of data, you might update exactly what the appropriate granularity is. If you have very little data, then you might coarsen a lot more because your data just doesn't give you more information about what's going on between the cause and effect. So I think this is a sort of nice standard to use because it allows us to not come in with our preconceived concept of at what level of granularity we should represent the cause and effect relationship, but to say that, okay, let's see what the data will tell us and then revise appropriately. So this way, I think we do have a technique for selecting among all the possible representations that we might have for the causal relations, ones that are sort of privileged. Of course, this sort of account depends on what do you measure and what sort of sample size do you have overall. Okay, so with that, then I want to turn to looking at actually implementing this sort of proposal for a theory of identifying the right granularity of cause and effect in terms of a specific study that we did with together with Ralph Adolph and colleagues at the University of Iowa. So the thought is, as I had indicated earlier, that we want to take lesion maps. What do I mean by that? We have, for each voxel in the brain, we know whether it's lesioned or not, okay? It's just a binary map with relatively high granularity for the patient's brain, and we just know where exactly that lesion is. And for all of these subjects that we studied, we had detailed answers to psychometric tests. And so the first sort of tests that I'm going to focus on were language tests and visual spatial tests. So what we had here are, for the first experiment or first analysis that I want to show you, we had about 500 individuals, and you see heat maps of the lesions in the first panel. These subjects had all sorts of different types of lesions, and the tests, the psychometric tests, were run quite a while after the lesion was occurred. So there's not sort of a directness, any sort of plasticity that would have happened that sort of sorted itself out by this. The histograms on the right-hand side indicate what their performance was on two different tests. One was a language learning, a language test, language ability test, and or word order test, and the other one was a visual spatial test. Both of these scores, in this case, were just scalar values. They were sums over a whole lot of individual tests that the individual. Okay, so these were our subjects. So on the left-hand side, on the cause side, we have brain maps that are just binary of where the lesions were for each of the subjects. And on the right-hand side, we're not going to do any coarsening or fine-graining there because we just have two scalars, namely how well did they do on the language test, how well did they do on the visual spatial test. Now, as you know, presumably deficits to language are often located, language ability is generally located in the left hemisphere, so we would expect deficits in language performance with lesions in the left hemisphere, and similarly for visual spatial performance, we would expect that to decrease with lesions in the right hemisphere. Okay, what did we do? We had developed this sort of method based on the principles that I had discussed before about finding the right granularity that we call causal feature learning. And there's a lot to be said about how exactly it works and what you should do in each case. But so here are the sort of general steps that we went through. We first actually tried to learn the high-dimensional mapping between the lesion map and the cognitive test scores. This is a very hard thing to do because you have a very high-dimensional input and a very low-dimensional or possibly high-dimensional output as well, and you have relatively little data, and you have to try to learn the sort of high-dimensional relation. Once you've learned that relation, you want to cluster the types of lesions, or the different lesions together for which you are predicting the same cognitive scores on the outcome tests. And you want to stop clustering at that point at which you would be clustering two different types of lesions together for which you have different predictions on the output. That is where you want to preserve the definiteness of the causal effect. Now, so far, there's nothing that distinguishes this from identifying correlations versus causation, but I'll come back to that in just a moment. Now, each of these steps you can ask, how are you going to do this? And how you do each of these steps depends very much on the domain that you're working in or the amount of data that you have. So, for example, the first step of learning the high-dimensional mapping, you might want to do in terms of a neural net, or if you have very little data, then you might have to just use a ridge regression or something like that. So this is going to be a function of what sort of data and what sort of tools you can actually apply. We did both, actually, and so both using a neural net and using a ridge regression, because we were sort of nervous that effects of the architecture of the neural net might be greater than what we're identifying. But in fact, we found almost identical results with the neural net or with the ridge regression. The second one is, how do you do clustering? Now, you don't want to cluster where you presume already in advance how many clusters you're going to end up with, because that would be sort of assuming a level of granularity already. So you want to have a clustering method where you can see, where you can test at what sort of granularity you want to stop. So that, again, it depends how much data you have and what you can do in that space. The code that we have is completely flexible in that regard. So what did we get? Here, this is a very busy slide, so let me talk you through what's going on here. So what we identified were 5 different lesion clusters. Those are the 5 rows of brains that you see on the left-hand side of the plot. And we have the heat map here that says where the lesions are predominantly for those people who fall into each of these clusters. Now, we could have ended up with 3 clusters. We could have ended up with 10 clusters. We ended up with 5 clusters to that were supposedly the right granularity of aggregation, given that given the data we had and given the sort of measures we had. So what do we see? We have the brains that give you some indication for each cluster what sort of lesions the people in those clusters had. We have in the B column, we have the size of the lesions for the people in that cluster, and then in the green panels on the right-hand side indicate how well did people in those clusters do on first the language task and secondly on the visual spatial task. So what you see is that perhaps unsurprisingly, in the first cluster, we identify lesions in the right hemisphere that correspond to a significant decrease in performance in the visual spatial task, but don't seem to have any sort of significant effect on the language performance. Whereas in contrast, the second category identifies lesions in the left hemisphere, for which we see a significant decrease in performance in the language task, whereas we don't see much change in the visual spatial task. In the third category, we also see left hemisphere lesions, but those lesions are much smaller, but they also result in significant language deficits. Strangely, what we find in the fourth category, and this was very, very disconcerting for us at the time, we see frontal cortex lesions, which result in an improved performance in language, a significantly improved performance in language tasks. You can't believe how long we searched for a possible drop of a minus in our analysis, because we don't want to go around recommending frontal cortex lesions to improve your visual spatial ability. Well, this is what we get in our data. When we talked to the University of Iowa neurologists, they said this is actually known to some extent, that certain types of frontal cortex lesions apparently can result in increased performance on some of these tasks. Of course, what this also means is that we don't know from the measurements that we have in the effect what sort of things that this part of the frontal cortex does that would be deteriorating in this case. Okay. What I find reassuring or sort of exciting about it is that we get this for free. We had no idea, at least the people who were actually working on the data analysis had no idea that there would be these types of lesions that result in increased performance, and sort of were surprised that this dropped out of the analysis. The last category, the fifth category, is a catch-all category. You see there's not much exciting going on in either of the task performances, and the distribution of lesions is all over the place in terms of the brain. So what's the upshot here? The sort of thought is that given the data that we had, the types of lesion distributions that we had among our 500 subjects, and the types of language and visual-spatial performances that we had in the test results, this was the granularity that we got out if we applied the sort of principles for identifying the right level of causal analysis that I had argued for before. Okay? Now, so the picture that we have here is that we didn't do anything in terms of finding the right level of granularity of the effect, because we just had the aggregate scores for both the language and the visual spatial test, but we did sort of try to find the right level of aggregation in the cause. Now, hopefully what at least some of you are thinking is that I haven't said anything about confounding yet, right? How do I know that the relation that I found between these 5 types of lesion categories and the visual and the cognitive task performance, why is that not due to confounding? Well, the first thing to say about this is that we're dealing with lesions here. So in some sense, you might say, well, lesions are sort of quasi-interventional. They're not a randomized controlled trial, at least not for these subjects, and I hope not for any subjects. But they are, in some sense, an intervention. Now, that doesn't mean that there can't be people who have had that there might be some sort of confounding, that certain types of people get particular lesions, and also certain types of people exhibit certain performances on these cognitive tasks. So we can ask ourselves, given that we can't actually intervene on these subjects, we can ask ourselves, what can we do to control for confounding? Well, one thing we can at the very least do is we can check for the demographics that we have for these people. So since that is one obvious source for where you might think that this sort of confounding could arise from, we did that. And the sort of thought was that, okay, what we need to check in terms of whether the demographics are confounding this sort of relation or whether the lesions are actually causal of this effect is that if we include the demographic information on the cause side and we see a change in the type of categories that are identified by our method, then that indeed would suggest confounding. In contrast, if we do not find any change in the sort of categories of lesions that we identify, even if we include the demographics, then that must mean that the effect of the demographics is screened off by the types of lesions that we observe. Okay, we did that. We basically ran the entire process twice, once including the demographics, once not including the demographics. This is a Sankey diagram that on the left hand shows the types of categories that we found that I showed you before, the 5 categories there. On the right-hand side, it shows if we included the demographics, what we had as demographics was age at scan, sex, and years of education. And you see that basically there was no change whatsoever, or minimal change in the categories. In principle, what could have happened at this point is that not only could there have been a large shift in the groupings in how the subjects are allocated to groups, but we could have also had different groupings on the right-hand side than we had on the left-hand side. So that was a possibility that didn't happen. Okay, so the upshot of that is that we feel reasonably confident that the demographics are not confounding our results. Of course, that doesn't mean that there can't be other unobserved confounders that might still account, that might still confound the results we have. We rely then in this case on the quasi-experimental or quasi-interventional nature of the lesions, and we're not going to do any interventions on these subjects. We might do some more analysis to see whether we can use other observational techniques to identify confounding, but we haven't done that yet. Okay. In some sense, that was so far just recovering information from the data that is already known in the field. So it was no surprise to anyone that left hemisphere lesions resulted in language deficits and that right hemisphere lesions resulted in visual spatial deficits. It was, at least to some of us, a surprise, certainly to me, that certain frontal cortex lesions seem to result in improved performance for these subjects. But it would be interesting to take a space where we don't know so well what the types of lesion categories are that might be causal of the outcome, of the effects. And so with these subjects, I mean, the University of Iowa has really this extraordinary database of lesion subjects. We could study something that was less well understood, I think. So on the effect side, we now have a BDI depression questionnaire, which consists of 21 questions. Each question can be scored, I think, from 0 to 4 or something like that. And so we have the detailed individual level answers to the questions on the effect side, and we have, again, lesion maps on the cause side, and we are interested in identifying what is the right granularity of the effect. So cohort, again, we've got a whole variety of different lesions as shown in the heat maps of the lesions in the panel A. In panel B, we have the responses of the participants to the 21 questions of the BDI and the correlation matrix between those. And these sorts of questions are also grouped into somatic and a cognitive dimension, and we have summed those up and provided the correlations for those in the second red correlation matrix. And then on the right-hand side, you see a sort of a bar chart of how the people, what the sort of scores were across our 520 subjects for each of the 21 questions. So what's the idea again? We've got lesion maps on the cause side. We've got the detailed, the individual answers to the 21 questions on the effect side. Now we want to go up on both sides. We want to sort of find the right granularity on both sides. Okay, what do we get? We got, as we turn the crank here, we only got 3 categories on the lesion sides. These are the 3 rows that you see here. And we got 4 sort of answer profiles, which you see on the right-hand side. Let me talk you through some of them. So what does this mean? The first category is sort of a generic category of all sorts of different types of lesions. And a lot of these lesions lead to, so the darker greens in the answers mean that the person is generally considered to be more depressed, lead to significant depression measures. The second category captures lesions. It's a little bit difficult to sort of visualize this because you have to visualize the whole brain and across a whole bunch of subjects. But here you see that there are lesions that are sort of what you call this dorsolateral prefrontal cortex or something like that. Does that seem right? So that really primarily lead to really negative outcomes. And then just as in the other set of, in the other cohort and with the other sort of cognitive outcomes, we have these sort of ventromedial PSC lesions in the front, in the third category, that is the only category that really leads to good outcomes, which is the top response category. It's sort of surprising. There's also a significant number who have bad outcomes, but the third category overall is the only one that leads to the good outcomes, whereas even the generic category in C1 doesn't have anyone going into the positive outcome. I should say the answer category E2 it doesn't have any arrows going into it. There are arrows going into it, but their threshold, there's a threshold of arrows that are smaller than 0.1. So what does this suggest is that we identify different types of lesions, but we don't get a sort of very clear pattern here of where exactly these lesions are. But we do, we are able to sort of distinguish at least 2 different types of lesions that seem to have very different effects. And now we can ask about the effect size. Well, why is this the right granularity? Should we be thinking in terms of measuring depression, perhaps more in terms of these 4 categories rather than in the aggregate BDI score? So just before I go and answer that question a little bit more, let me say we did a check on demographics here as well, and for possible confounding due to demographics, and we didn't find any effect here either. So One thing that we have spent quite a bit of time on, I see, I skipped those slides, is we looked at whether these four answer categories are actually better representations than either the somatic and cognitive dimensions for measuring depression or for the aggregate BDI score, total BDI score as answer. It turns out that there's really not much difference in terms of the quality of those representations, but here you have just four categories rather than a continuous score. So that suggests, at least from these 21 questions, the aggregate BDI score is actually pretty good, but these sort of four categories would be the sort of natural thing that you would get out if you took the principles that I advocated for at the beginning for identifying the right effect granuli. Okay. Now, I showed this already. We don't know the ground truth. Maybe you don't believe me that this works. So I'm going to try just one last time. We did it with simulations as well. So in this case, we constructed deficits. We took the lesions that we had among our subjects, but we constructed the deficits and we constructed what sort of performance those deficits resulted in terms of made-up cognitive tasks that are the green outputs here. So what you're seeing is we take lesions in the across the brain, we flip them all to the left hemisphere just so that we have more data on the left hemisphere, and we have three deficits that we're interested in, those three that are in green there, and they are caused, this is now ground truth, by lesions that overlap with those areas here. Okay, that's the sort of idea. And we've got a model for how we relate the two. Now, an interesting question would be is that, okay, how would others do this sort of analysis? One sort of way that is standard in the field is to use canonical correlation analysis, where they map the brain map, where they map the lesion map of the, where they map the lesion of the brain through a bottleneck, a sparse bottleneck, and then have a linear encoding of the answers to the questionnaires in terms of that sparse bottleneck. One way to think of canonical correlation analysis is really that it provides a dimensionality reduction of the lesion map, and it provides a dimensionality reduction of the responses, and then tries to find a sparse mapping between those two dimensionality reduced spaces. Now, if we do that on the simulated task, and we actually tried a bunch of different techniques, and there are then a whole bunch of different measures that you might look at of how good the outcome is, then we are the causal feature learning column. Now, in the first plot, you want to be as high as possible. In all the others, you want to be as low as possible. What you see, and these are different measures of how accurate the delineation of the true lesion is given different methods. LESI map is actually a canonical correlation analysis-based method developed by our colleagues at the University of Iowa. It does very well across the board, but we are exactly with them, or more or less exactly with them in most of these. In some cases, we're a little bit worse. In some cases, we're a little bit, are we better? I'm not quite sure we're better in any of these. So this so far only suggests that we're sort of competitive with other methods. But one of the things that really is disturbing about the canonical correlation analysis results is that how do you interpret them? So if you look at the output of what would otherwise be used, namely canonical correlation analysis for this sort of task, then what you get is that, okay, here are the three deficits that are identified. The location of those deficits is actually pretty good, as the previous plot showed, but now it's really unclear how to understand them. So the first deficit is good. It says that, okay, it makes lesions in this particular location make that cognitive skill worse. But the second and third one are really confusing because they say that some aspects, some lesions in some parts make things worse, and lesions in other parts make things better, namely blue or red. And we know from the ground truth that in all cases the lesions only make things worse. Okay, so one of the really difficult parts of analyzing this sort of output is to sort of interpret of what actually goes on. In contrast, if you look at our results, what we get from this sort of simulated data sets, we got 6 categories out. Now note that there were 3 types of lesions, but if you look at, so the accuracy of the location of those lesions that we identify is spot on, or very close to spot on. Now, what you see is if you look at the first lesion, it exactly nails deficit A, and you can see deficit B and C are hardly affected at all. It just captures the people who have deficit A. For the second one, it's deficit B. We clearly identify that lesions in that area are have an effect on skill B. And similarly for C, in the third category, you see that the lesion that is identified in the third category accounts for the skill deficit of ability C. Now, if you look at category 4 and 5, what you actually see is that this captures lesions, this captures people who had large lesions that covered several deficit areas. And so in the case of category 4, it's lesions that covered the areas that would both influence deficit A and deficit C. And then in the case of the category 5, it's lesions that influenced all 3 skills. So in some sense, we capture even the sort of interactive effects if someone has a large enough lesion that affects 2 skills, and pull those out as the right category. The last category is the catch-all. It's sort of generic lesions that don't have any sort of well-defined effect on any of the individual features. Okay. Now, I hope that provides some sort of evidence for you that if we do know ground truth, what we recover with this causal feature learning method is not only the accurate location of the lesion, but in a very interpretable way where you can really see that, okay, this sort of lesion in this specific area affects that sort of deficit, and that even if lesions overlap multiple areas, that we sort of pick up that sort of feature exactly as we would expect. So we were very heartened by these results. To be honest with you, I didn't expect them to come out so nicely. So as a conclusion, then, this sort of method that we've been working on, I think, offers a really systematic approach to finding the right sort of categories on both the cause and effect side, given high-dimensional and multimodal measurement data. I think that's sort of exciting for many, many domains, not just for neuroscience. The granularity of the representation is in all cases empirically defined. So it's not that you come in with your sort of concepts of where the right types of lesions are to knock out the language ability, but this is the sort of thing that you are going to start identifying and defining by this sort of analysis. But obviously, this sort of empirical result is highly dependent on what sort of data you have available and to what extent the measurement space that you used was appropriate. So, for example, in the depression case, we just had the 21 questions to measure depression. Maybe it would be interesting to have video, detailed video of people acting in a more natural environment. Maybe we would find other sorts of relevant features that are effects of lesions that capture the relation between lesions and depression symptoms. And so I think both the simulations on the ground truth, where we knew the ground truth, and the recovery of the known lesion symptom mappings in the case of language and visual, in the case of language deficits and visual spatial deficits, I think mean that we can be in business. And so I have actually some confidence in the results for spaces where we didn't know the answers already. Thank you.
I will take the chair prerogative to ask the first question. You mentioned some confounders, and I was wondering if you can imagine a confounder where somebody has perhaps a larger language area which could affect the likelihood of having a lesion in that area, as well as affecting their ability. maybe that's not very likely, but you could potentially test for something like that in simulation. So I was just wondering if this is something you've ever thought about.
Oh, okay, so help me out. You have a large language area, but the lesion only hits a small part of that, or what's the?
If you have a large language area and you have random lesions,
Yeah.
then you're more likely to hit at least part of that area.
Yeah.
But then also, if you have maybe a larger language area, maybe have better language abilities, so it could affect both sides of the.
Yes. So okay, good. We have not actually gone through the individual subjects to see like how large is the lesion compared to what is commonly thought to be the language area. So, but I guess the sort of question is like, where would you get the knowledge from of how large their language area is?
No, but you could test that in simulations potentially. That's
Sure.
Yeah.
That's simulations. Yeah.
So, sorry, well, I was ignoring this side before, so let's start with Bill, then Aaron, Adina, Megan.
Frederick, I'm really impressed with the methodology, the rigor and elegance of it. And I'm then astounded that at the end of the day, the main effects you show are exactly the ones that were shown by the split brain studies back in the 60s and 70s. You know, language in the left and visuospatial localized to the right. And I've been teaching this to Stanford medical students for 30 years, resolutely ignoring the regular things that pop up in the literature about right hemisphere specialization for holistic thinking and, emotional relations, and left hemisphere for analytic and blah, blah, blah. So I'm curious about your questionnaire and how well you parsed the space of possible effects. For example, I would have expected some posterior cerebral artery infarcts to have vision deficits and that to show up in your analysis, unless you just didn't consider anything about vision. I would have expected some motor cortex deficits. They should be very large with these big middle cerebral artery, but if that's not included in your analysis, obviously it's not going to pop out. So I'm kind of wondering, how detailed were those questionnaires, and are you limiting your clusters and your findings based on not sampling the space of possible data you could have gotten from the subjects? Am I clear?
Yes. Well, let's see whether my answer addresses it. So, of course, there are many, many details that we're missing, but so we have to work with what we've got, right? So, we didn't go back to search for particular lesion subjects. We had a database of lesion subjects for which we had some test results. What we happen to have in terms of test results were, for these subjects, the COA and the JLO, the line orientation task, since you're asking about visual spatial tasks, right? So an obvious question is like, well, I wish we had like a whole bunch more visual tasks, and I wish we had, well, perhaps I don't wish for more lesion patients, but you know what I mean.
I know what you mean.
Is that we don't have perhaps the distribution or the coverage of different types of lesions to allow us to identify other areas or more specific areas with regard to the visual task. So this is where I think the second bullet here is absolutely crucial, right? Is we had 500 subjects which had the lesions wherever they were. That's the subjects were not selected for having lesions in particular places. That's in part the point, right? Because we don't want to say that let's take our hands off in presupposing where the lesions ought to be in order to see the deficits, right? So maybe we could go back and look at the individuals and see whether there are specific ones with specific types of lesions that then and see where we categorize them or why they don't pop out. But actually my worry is that we are at the absolute minimum of sample size already to do this sort of inference. And so we were sort of scrambling with many, many ways to sort of even be able to turn the crank, given that we had 500 subjects. Now, in contrast, I think that if we were working in a space where sort of sample size was not an issue, that would be absolutely easy. So I think fMRI, for example, could be a sort of interesting case where we'd have oodles of data on the cause side and on the effect side, and then we could turn a crank that has perhaps then much more fine-grained results. But I think in some sense that's the right result, is that given that we only had so few samples, our results should be pretty coarse. And so that's what it is, I think.
Yeah.
Can I just make a quick comment to Bill's? I think the specific cases that you mentioned, Bill, would be excluded because they would have been considered confounds. If you have a patient that has a visual field defect, you wouldn't test them on judgment of line orientation because they can't see well in the first place. They would be excluded from doing that task. Similarly, aphasic patients would be excluded from doing the language task, et cetera. So I think some of those cases that you imagined, they would cause deficits, but those would be considered confounds, and they wouldn't have been administered the task in the first place for that reason. Does it make sense?
Well, it makes sense, but I would consider, you don't test them on the orientation because they report visual field deficits. It's not a confound; it's the main effect.
Well, I mean, I agree it's not a confound, but I mean, they might still have been excluded from that test, and we didn't have a choice about that.
Yeah, I understand.
Was Ralph right about excluding all the aphasia patients from the language test? That seems a bit extreme.
It depends on how aphasic they were, but yes, if they're severely aphasic, they would be excluded because they wouldn't be able to produce output.
I think it was Aaron, then Adina, then Megan. Yeah.
So my question is about the causal. You mentioned earlier in your talk about the causal relationship between the brain and action and behavior. If you start at the neuromuscular junction and for a given muscle contraction and you follow synapses backwards, things diverge massively. They don't converge. And so how do you deal with cases where the relevant cause is a state, not a place?
I can't deal with them in this data, right? Because I just have, remember what I have as input. I just have whether or not this part of the brain was lesioned. So this data would not address that question, right?
Sorry, I didn't mean in the context of this data, but in general, in terms of the methodology.
Oh, so but then okay. Sorry, I misunderstood. So yeah. Then the question is, what is the input data? So if I had activations in the brain, right, so detailed activations, then I could start working with those, right? And I could say that, okay, what sort of activation patterns would I expect to see such that, whatever output you want to measure.
So you can still use this approach where you intervene. That just seems really tricky.
Say more, because perhaps I'm just misunderstanding the question.
So, my understanding of the idea is that you're looking for your ability to control, right? That if you can isolate and then control, that's the cause. But in the case of where the cause is a state rather than a place, it's very hard to isolate.
But so the claim would be, maybe we'd have to talk through exactly what the setup is. The claim would be, if there were activation, is that I would be able to say, okay, you would have to have this sort of change in activation in this area such that you see that sort of effect. That I could still do. Right? Mm-hmm. I'm now not sure whether that actually addresses your question.
I'll check with you offline.
Yeah, I think my question is sort of a follow-up on Bill's, but maybe I didn't quite understand. With respect to the effects, the tests that you were using, were you using aggregate scores on tests as opposed to sort of more fine-grained test answers for these?
Okay, so I did both, right? So in the language and visual spatial one, we only had, that was the only thing that was in the data, the aggregate scores that I used. That is basically the sum over all the questions. So we had two scalars as effect in the first results I showed.
So that's really why you didn't find anything more fine-grained. It's like you didn't give it anything fine-grained, but had you had the specific answers across all of the batteries of tests to go along with the lesion maps, you might have found, let's say, the cognitive ontology these kinds of lesions in terms of the patterns of effects that were picked out as being salient. Is that what?
So okay, we did put in the two tests at the same time, right? So in that case, and so of course what we wanted was like every single test that we could get, right? And ideally we would want it at the finest granularity that it was measured. That was not available. So you're right, is that we use the aggregate scores, but I guess what I want to say is that using the aggregate scores is actually the common way that people do this sort of lesion behavior mapping. And when we did it for the depression, where we actually used the individual answer, I mean, this is like you scored 4, or you rated 4 on question 3, right? So that goes in then, and then we get this sort of categorization into 4 effect states and 3 cause states in the brain. I still think that, you know, given the sample size, we were sort of, well, we were limited mainly by the data in the first place and what was measured. But yes, if we had many more visual spatial tasks, then maybe we would pick up these sorts of different types of locations for the cause. But it's not obvious to me that if we had a lot of data, that we wouldn't pick up some of them also by just using the aggregates. That I think is an open empirical question.
Mm-hmm.
Yeah, you know, I'm sorry.
Megan, sorry, I think Megan is
If it's a follow-up on that, then I can wait.
Let me try it quickly. It came to me in that context.
No. Okay.
It is a follow-up in the sense that you talked at the beginning about, you know, not making sense to you to do a sort of like a components or factor analysis within each of the domains. But of course, the fact that you didn't find anything in the depression scores might be due to the fact that there isn't anything besides like a single, you know, there's really just one common factor underlying all of those 21 questions, right?
Absolutely. So, I mean, I think actually that's our problem in the depression one, is that BDI, so the depression score has been studied so carefully and it's so well tuned that we're just picking up one feature, basically, and all those questions work towards that one feature. And so actually we looked at the sort of detailed analysis. Every question adds this a little bit to it. And so it's not even that we drop out questions in a significant way. But then the upshot is that the problem is we're not measuring enough in the effect. And we should look at a broader, like we should ask a bajillion questions, right, if we could, and then turn this crank, and then we might find something more interesting. Yes, so I completely agree.
Good. I'm glad I waited, actually. So my question has been touched upon by a couple of your answers, actually, which is you said, well, you know, if we had functional data, then, you know, maybe we could do something more interesting. If we had more complex behavioral outputs, maybe we could do something more interesting. And there are a lot of cool functional data sets out there where you have like 10 hours of data per person as they're like driving around in a virtual environment or something. And so my question is really, what are the considerations for taking this method and integrating it with a functional pipeline or with a pipeline that takes in as input something other than structural and lesion data? So not just beta weights that come out of a GLM or even just raw bold signal, but something like functional connectivity or dynamic functional, to the extent that you think that dynamic functional connectivity is a meaningful signal. Anything like, is this method agnostic to a certain degree to what you put into the pipeline? Could you integrate this with functional pipelines and then go after all of those complex behaviors for data sets that exist out there in the world?
Well, we started with El Nino when we first developed this method and used satellite data over the equatorial Pacific and to identify, to sort of learn whether we could automatically identify El Nino, which we did, and the contrast La Nina as well. And we've used it on trade data and economics, where we're interested in like what are the sort of trade features that result in long-term growth. So the method, the sort of principles that I specified at the beginning are completely domain general. The challenge in sort of functional data or imaging data is like, okay, what are we going to use actually as data input, and what is the sort of effect space that we're measuring, right? What are you going to use? And then just getting the person who really knows how to handle that sort of data and turn the cranks with these sorts of methods. So, but yes, the method is completely domain general.
That's cool. Just to follow up really briefly, I know that Champagne is waiting, but to say that some of these data sets also could mean that you could do this like multiple stages. So you would have not just brain causing behavior, but you would have stimulus causing brain causing downstream behavior. And so some of these like naturalistic driving environments that like Jack Gallant has, for example, they've got massively annotated inputs in the visual, like they have just for their encoding models, stupid amounts of features that they have gone and actually painstakingly developed on these stimuli. And then they know where the people are looking, and then they have the brain response and the subsequent behavior that the participant engages in. So it feels like there could be some really fun ways there to take this so that it's not just cause and effect, it's cause and effect, which then becomes cause to create behavior. And that could be a really interesting way to go.
You're preaching to the choir. So yes, absolutely. But I mean, so Ralph and I have discussed this with regard to, video stimulus and then brain measurement, brain data measurement as effect, right? So how do we do that? As the input becomes high dimensional and the output becomes high dimensional, the sample size demands explode, right? So sure. And then computation demands explode, and that means demands for good programmers explode. So
thanks.
Let's thank the speaker and then stick around for a moment. Thank you again.